And I'm gonna turn off my video at this moment. And okay. Okay. Morning, everyone. Let's get started. Okay, so last time we mentioned Morgan's very famous Morgan's experiment on a fruit fly. So in this experiment, he identified an important gene. So this gene, let's say W plus, is the wild type. This allele will produce the red eye for the Drosophila. And there's a mutant allele, W, without the plus sign here. So that allele will produce the white eye in Drosophila. So the wild type is dominant to the mutant type. Okay, so in his experiment, the first cross, Morgan crossed the red eye female with the white eye male. Now everyone should be expert in using the Punnett square, right? So if we use the Punnett square or Punnett rectangle to dissect this cross, so this will be the F1 progeny. So as what is expected. So in F1, everyone is red eye, right? Then when Morgan did the intercross among this F1 progeny to get the F2 population. So in this F2 population, he observed like 3/4 of the F2 progeny show the red eye, and only 1/4 of the F2 progeny showing the white eye, which is expected. However, so when we look very closely at this results, you're gonna see all the white eye progeny, F2 progeny, are male. So this is very interesting. So then he did another cross, the second cross. In the second cross, as shown here, Morgan just exchanged the role of the parents. So this time he crossed the white female with the red male, and in F1 progeny, he immediately observed the segregation of the phenotypes. So half of the F1 progeny showing the red eye, and the other half showing the white eye. And all the white eye F1 progeny are male, and all the red eye progeny, F1 progeny, are female. So this is interesting. And then he also did the intercross between this F1 progeny to produce the F2 progeny. So as you can see, half of the F2 progeny are red eyes, and the other half are the white eyes. And within the red eyes, half of them are female and half of them are male. And this is the same in the white eye progeny, F2 progeny. So by doing this, Morgan was the first one to demonstrate that genes and traits are linked to chromosome, and the DNA's genetic materials are located on the chromosome. And he won a Nobel Prize for this important and famous discovery. Right. Okay, now we're going to learn a very important concepts here. So aneuploidy. So let's look at the euploidy first. Euploidy means the standard or normal chromosome setting in a cell or in an organism. So for example, this is a standard chromosome setting in a normal person as you can see, we have there are like 46 chromosomes in this cell, right? So this is called the euploidy, and the aneuploidy means non-standard or abnormal chromosome setting in a cell or in any organism. This is an example here. All right. So if you compare the first one and the second one, the difference is here, right? Because in the second cell, you're going to see 3 copies of 21 chromosome. So this case, so for this situation, we call this situation as a trisomy 21, which means this cell, this individual has 3 copies of chromosome number 21. And this abnormality will cause different various phenotypes, for example, the growth failure or the mental retardation. All right. So this is a very classical human disorder. Okay, so as I mentioned, euploidy. Euploidy means the normal setting for the chromosomes. Either one set of the chromosome haploid, 2 sets diploid, 3 sets triploid, and so on. So all these are called the euploidy because they are normal setting. However, if in any cell, as I mentioned, in any cell or in any organism, if you see like one entire chromosome missing or one entire chromosome extra, so those are the non-standard situation. We call this as the aneuploidy. The first one of this type is called the trisomic, which is the one entire chromosome extra. We also can say the 2n+1 because the 2n is the standard, right? The euploidy and has 1 chromosome extra. So if in any individual or in any cell, if you see a entire chromosome missing, then this will lead to the 2n-1 chromosome. So 1 is missing. And in this situation, we call this cell or this organism individual as a monosomic. So this is an example. So for example, if a gamete, this is a gamete. If one gamete has an extra chromosome, when this gamete come together with a normal gamete to form a zygote, so this zygote will have an entire chromosome extra chromosome. So this will lead to the 2n+1, which is the trisomy. And similar, so if one gamete has 1 chromosome missing, so this individual, the zygote will be the monosomic. So how and why does this happen like this? So how to get this kind of abnormality in chromosome setting? So one possible way is through, oops, let me erase this first. Okay, so one possible way to cause this either the trisomic or monosomic is through the non-disjunction. So what is the non-disjunction? All right, the definition of the non-disjunction is the failure of either the homologous chromosome or the sister chromatids to separate properly during cell division. So let's look at these examples here on the left. So during a meiosis, in the anaphase 1, as all of us should be able to, as all of us should know what is happening in the meiosis, right? In this phase, so the 2 homologous chromosomes should be paired together, and all the homologous chromosome pairs are lining up along the cell equator being ready to separate, right? Okay, now if something goes wrong, so if it is a normal situation, so these two homologous chromosomes will end up in two different daughter cells, right? This one should go to this cell, and this one should go to this cell. So that's the normal situation. However, if something goes wrong, and these two sister chromosomes do not want to separate, so they always come together. So what would happen? So these two homologous chromosomes, these two homologous chromosomes, if they went to the same daughter cell, this will lead to what? The consequence of this is like one daughter cell will have two copies of this chromosome, but the other one has none of this chromosome. All right. Now, after the second division, you're gonna see in this meiosis, all the gametes will be aneuploidy, right? Half of them will be the trisomy, something like that, and the other one will be, so half of the gametes will have an extra chromosome, and the other half of the gametes will have one chromosome missing. All right, so this is one way to produce the non-standard gametes through the non-disjunction. And this is the example showing the non-disjunction occurring at the first division. And this situation is very rare. So it's not likely, but it's possible. That's why, we do not see this very often. It's a rare situation. All right, this is the first possibility. So second possibility is that the non-disjunction could occur at the second division. So the first division is normal. Two homologous chromosomes, they are correctly end up in two different daughter cells. However, in one daughter cell, so the two sister chromatids do not end up in two different daughter cells. They come together and join into the same cell. So this will produce a non-standard gametes and another non-standard gametes. So this is the mechanism of the non-disjunction, which can produce the non-standard gametes. And this non-standard gametes can produce either the trisomic or the monosomic, the aneuploidy individuals. All right. So last time we mentioned there's some like non-standard the sex chromosome setting in human. So this, so this kind of like sex chromosome setting, either XO or XXY, those situations are caused by those non-standard sex chromosomes makeup are the product of non-disjunction meiosis, right? So I hope this is clear, right? Okay, so let's look at the famous Morgan's famous experiment again. So as I mentioned, yep, go ahead.
Sure, I had a quick question. So for the previous like first division, second division, you said one is more likely. Is the first division what occurs most often when you see non-disjunction?
Oh, sorry. So I think I need to say that. So all this kind of like non-disjunction will be rare situation, not just the non-disjunction at the first division or at the second division. So both are rare, right?
Okay, got it. Okay. So okay.
Any other questions? Yeah, thank you for pointing out. Yes. So so when I talk about the first division, so I said this is a rare situation, but I didn't mention if non-disjunction occurring at the second division, it's also rare. So the entire situation is very rare. So that's why we see a very rare cases, like for example, the trisomy 21, right? This is a very, very rare human cases in human population. All right, thank you. Okay, so both situations are rare, and this can cause the aneuploidy, and the non-disjunction could occur at the first cell division or at the second division during the meiosis. Right. Okay, so let's look at the famous Morgan's experiment. All right. So in this experiment, Morgan crossed a red-eye male with a white-eye female, and this is the standard output, right, from for the F1 population. So if you look at this Punnett square, you're going to see. So the progeny, the F1 progeny from this cross, half of them are the red eyes, and the other half have white eyes. However, all the white eyes are male and all the red eyes are female. So this is why. This is because this gene is located on X chromosome, right? So because we knew this gene is linked to the X chromosome. That's why. So the inheritance pattern will be influenced by sex. All right. So he did a lot of experiment to prove this many times. Always he got this standard and normal outcomes. However, in a very rare situation, so he got this. This is very rare situation. He used the same parents here, the red-eye male and the white-eye female. He used the same parents and did the cross. And in a very rare situation, what he got for the F1 progeny is half of them are white and half of them are red, but the red are always male and the white are always female. So this is totally opposite with this, right? So if you compare these two crosses, the second crosses are completely different from the first one. So why is that? This is because of the non-disjunction, right? Let me erase this. This is too messy. Okay, I hope, yeah, it is clear. So let's look at this very carefully, because, okay, let's look at this female individual, white-eyed. So when this individual produce gametes in a very rare situation, which means like the non-disjunction occurs, so this individual will produce two types of non-standard gametes. So one gamete with two X chromosomes here, and another gamete has no X chromosomes here. So this is a very typical classical non-disjunction occurring in this case. All right. Now, if this gametes with two X chromosomes form a zygote with a Y chromosome here, so this will produce. Why this produce a female fruit fly? So I hope everyone understand this, because this is very important. Because in tomorrow's quiz and the next week's midterm, I'm gonna definitely test you on this knowledge. So in fruit flies, what's the mechanism for us to use to determine sex in fruit flies? And in human, what is the mechanism for us to determine a male or a female? All right. Okay, so now.
So what you're saying?
Yeah.
Sorry.
Go ahead.
I was just gonna say. So what you're saying here is that a white-eyed female with that extra Y chromosome was still fertile and still viable, but a red-eyed male that was missing its Y chromosome was sterile. and
Oh, you mean this
every
one,
other
right?
iteration
Yes.
died.
Yes. So those two cases cannot survive. So that's why. So you do not see any of them, this kind of like genetic makeup in the F1 progeny from this class. Yes.
Okay.
All right. Okay, let's focus on this one. So this one is a female. Even there's a Y chromosome, right? This is not human. This is fruit flies. So we need to be clear, right? Because in fruit flies, we use the ratio of what? We use the ratio of X chromosome to the autosome, right? If this ratio equals to 1, so that will give you a female individual here. So when we look at this individual, so the ratio of X to autosome equals to 0.5. So this case, you will get a male individual. So that's why how we understand the difference between these two outcomes from these two processes, right? And this is the standard. So if you repeat Morgan's experiment for 1,000 times, probably like maybe 999 times, you got this standard output. But yeah, sometimes you got one case like this. So this will give you a surprise. All right, but if you understand what is going on behind, you're not surprised at all, right? So this is because of the non-disjunction occurring in this female parent. So any questions? I'm gonna ask you questions because I need to make sure everyone feel comfortable with this. Okay, very good. Okay, so yeah, so again, this is important. So how what information we need to use to determine either male or female in fruit flies. So this is criteria we use, right? We use the ratio of X chromosomes to the autosomes to determine sex in Drosophila. So why is that? Because so this is the mechanism. So this is not required in that in exams, but yeah, for your own benefit, I'm going to show you this slide. So okay, so those genes are located on X chromosomes, and those genes are located on the autosomes. All right. If the ratio is equal to 1, and so that means like the product from this X-linked genes is equivalent to the product, the amount of the product from this genes which are located on the autosomes. Then this pathway will be open, which will lead to the female phenotypes. However, if the ratio is like this, so 0.5, right? And so the gene product, the ratio of the gene product has been changed. Then this pathway will be blocked. So this pathway will be blocked. Only this pathway will be open, and this will lead to the male phenotypes. So this is the mechanism behind. All right. However, so in a standard situation, a female fruit fly has 2 copies of the X chromosomes, right? And the male fruit fly has 1 copy of the X chromosome. All right. So on the X chromosome, there are some genes that are very important to both sexes. They need to be expressed at a equal level in both female and male. But as you can see, male only has 1 copy of X chromosome, which means like male individuals only have 1 copy of those genes. But female has 2 copies. So then there will be a imbalance between male and female, right? But in Drosophila, there's a mechanism called the dosage compensation. Dosage compensation. So this mechanism will remain. Oops. Okay, dosage compensation, and this mechanism will accomplish the equalization of gene expressions between males and females of the Drosophila. All right, let's look at how this happens here. So this is a very famous example here. So Muller did a very interesting and famous experiment here. Okay, so let's look at the first female individual here. All right, so first, let's look at the genes that Muller has been working on. So this gene is W wild type. So this WA will give the fruit fly a red eye phenotype. So even one dose will give you something, right? So the pink color here. All right. So in this female individual, there are two X chromosomes. One X chromosome carries a wild type allele here, and the other one has a mutant allele. So this one will give the fruit fly a white eye. So one dose of this wild type allele. All right. Now, when he studied the second female here, so this female has two X chromosomes, and each of this X chromosome has a wild type allele. So two dosage here. So what is the consequence here? If you compare this one with this one, so if you compare this one and this one, so the two dosage will give you more redness than the first one, right? The one dosage. All right, one dosage here. So the two doses will give you more redness than the one dose. All right. Now let's move on to the third female individual. So for this one, this is a little bit interesting here. So this one has two complete intact X chromosome, each of which has a wild type allele. So two doses, right? And he also inserted another chromosome into this female individual. So this chromosome is called the Deletion X Chromosome. Why? Because it is not a complete X chromosome. It's a small portion of the X chromosome. So it has a very small portion, only contain this allele. So this will give this individual a third copy of this wild type allele. However, this extra X chromosome, the deletion X chromosome, will not contribute to the sex phenotype. So in this case, we also calculate the X to A ratio equals to 1, right? Okay, so this is a very special X chromosome. All right, now for this one, this female fruit fly has 3 doses of this wild type allele. See what happened here. So you're going to see the increased the phenotype, right? So it's even more red when compared to the 2 doses and the 1 doses here. Okay, this is what we expect to see, because more gene copies means more gene product, and so that means like those 3 copies will produce more red pigment than the 2 copies or than the 1 copies. However, when we look into the male fruit flies, something interesting happened here. Okay, so let's look at the first male. So this one, the male has one copy of the Y chromosome and 1 X chromosome. This X chromosome only has 1 copy of this allele, which means 1 dose. However, this male individual shows the same phenotypes than the female with 2 doses. All right, so 1 copy in male gives you the equivalent phenotype as female, which has 2 copies of this alleles, right? Okay, so he did another experiment. He looked into a second male. For this one, so this male has 1 normal X chromosomes with one dose of that allele. And this one also has a deletion X chromosome, so small, very tiny X chromosome carrying another dose copy of this allele. So this is 2 doses in male, but this one gives you a phenotype even which is even more rare than the 3 doses in the female. So what happened here? What happened here? So which means like there's a compensation, dosage compensation between male and female, right? Is that clear?
Yeah. Are you trying to say that it doesn't really matter how much of this how much of the mRNA or how much of the X chromosome is in that there is in an organism there, as long as it's determining something important like eye color in this case, it'll still express itself? Or
so yeah, that's a good question. So here I'm trying to say, so the dosage composition, what does that mean? That means like, okay, there's a mechanism that try to keep the balance between male and female, right? Because female usually has more copies of genes on the X chromosome than the male. So there's a mechanism that can enlarge the effect of these genes in male than the female. So for example, if you look at the first male, so this male only has 1 copy of this gene, but this 1 copy shows an enlarged effect than the single dose in the female. So so this 2, so let me remove this. Okay, so this female and this male showing the same phenotype because they are on this axis, right?
yes,
They're showing the same phenotype. But if you look at the genetic makeup between this female and the male, this male only has 1 copy of this allele, but the female has 2 copies. So which means 2.
hello.
Hey, Professor, you're cutting out. We can't hear you.
Yeah.
I'm sorry. Yeah, my internet was broken just now, so sorry about this. Let me continue. Yeah, go ahead.
Oh.
So sorry, I didn't finish your question, right? Answering your question. Yeah.
Yeah. So at this point, I do I think I understand what you're saying. What you're saying is that males, or at least whatever gender inherits the the like two different types of chromosomes, sex chromosomes, right?
Yeah.
They compensate. They have a mechanism that compensates for the lack of chromosomes that they have.
That is true.
In comparison to the other gender.
Mhm.
Okay, got it.
Okay, great. All right. All right. So okay. So all right. Let's move on. All right. So last time we mentioned how we can determine male or female in human, right? So this is the standard we're going to use, criteria we're going to use to determine sex in human. So we're going to use the presence of the Y chromosome to determine the male is or female is, right? The reason why is that because on Y chromosome, so this is X chromosome and this is Y chromosome, there's a gene only on Y chromosome. It's called the SRY. So this gene will determine the sex. So that's why in human, we use a different mechanism to determine sex. Okay. And in human, non-disjunction can occur in human to determine the sex as well, not the trisomy 21, right? So the non-disjunction will also be able to determine, to produce the non-standard sex in human. So for example, if in this female, if non-disjunction occurs, this non-disjunction will produce two types of non-standard gametes here. And this will produce the non-standard individuals with non-standard sex chromosome settings in human. All right, so dosage compensation. So I believe so we have covered the dosage compensation in fruit flies. So let's look at the same dosage compensation mechanism in different animals. So we have covered in flies. So in flies, this is the mechanism, right? Because in female, there are two X chromosomes, but in male, there's only one X chromosomes. So how we can fulfill the dosage compensation between male and female? So in fruit flies, this is the mechanism. So any genes, any X chromosome linked genes in male will be doubled. The transcription of any genes that is located on the X chromosomes in male will be doubled. So you're going to see a 2 times, right? 2X transcription rate in a single in the male. So if we double the gene expression on the X-linked genes in male, so this will be equivalent to the 2 copies of these genes in a female. So this is the mechanism for the dosage compensation in flies, Drosophila. All right, so let's look at the mechanism in C. elegans. So for example, in a worm. Okay, so in a worm, all right, the female has 2 copies of X chromosome. The male only has 1 copy of the X chromosomes. So to achieve the balance between the female and the male, so the worm in the worm world, they use a different mechanism. So what they use is try to suppress the genes the X-linked genes in female. So for example, 2 copies of the X chromosome in female, right? If worm can repress the gene expression on the X chromosomes in the female, then the 2 copies of the female X chromosomes will be equivalent to 1 copy of the male X chromosome. Is that clear? So if you compare the worms with the flies, they use 2 different mechanisms. In flies, they try to enhance the expression of the single copy in male to achieve the balance between the male and the female. But in worm, the mechanism is different. They try to reduce expression levels of X-linked genes in female to achieve this balance between male and female. So in mammal, like in human, we use another different mechanism to achieve the dosage compensation to achieve the balance between male and female. So what is that? Okay, so in human or in other mammals, so males only have 1 copy of X chromosome. Female has 2 copies. So how to achieve the balance between male and female? So the mechanism is called through the bar body. So in the female, there are 2 copies of X chromosome. So 1 copy will be randomly selected to be suppressed, all right? So this X chromosome will be wrapped and will be tightly wrapped using some mechanism. We're going to cover this shortly. So, and such that the genes, many of the genes on this wrapped X chromosome cannot be expressed. So what's the consequence? The consequence of this is like only the genes on this X chromosome can be expressed. So by doing this, this can achieve the balance between male and female. So in human, in mammals, so we use the actually we hyperactivate 1 X chromosome in each cell of the female to achieve this balance between male and female. Any questions here? Okay, so let's look at the bar body. Okay, so this is the mechanism of the dosage compensation in human or in mammals. Right? So in female, any female, there are 2 copies of X chromosome, right? On each of the X chromosome, there's a gene called the X-cyst. Right. So we have 2 copies, and in each cell, so 1 X chromosome will be randomly picked by fate. So for example, this one has been selected, and this gene on this X chromosome is going to be expressed. And the product from this gene expression is something called the X-cyst RNA. And the function of this RNA is to cover this entire X chromosome, to wrap this chromosome and try to condense this chromosome. So to form this bar body, this is called the bar body. And as a consequence, many of the genes on this X chromosome, the bar body, cannot be expressed. Only the genes on this intact X chromosome can be expressed. So 1 X chromosome can be functional. And this is equivalent to the male, right? In a male cell, there's X chromosome and there's a Y chromosome. So 1 versus 1, the balance between male and female has been achieved. Right. So this is the evidence to show the bar body in a cell. Any questions? All right, this is a very, very interesting example to show the bar body in mammals. All right. Okay, so probably many of you have cats. All right. So there's a gene on the X chromosome called the B, the capital B. The capital B gives cats the black hair, right? So the genotype of this X capital B and X capital B will gives a female black hair cat, and this genotype will give a male black hair cat. All right, so the mutant allele, the lowercase b here will give you a orange hair female, and this will give you a orange hair male. However, so when we have a genotype like this is a heterozygous, right? Heterozygous female. This shows the phenotype like this, the calico cats. So why is that? So if ideally, like the heterozygous like this will gives you a black hair phenotype. Why? Because the big B is dominant to the lowercase b, right? However, so why this cat can show phenotype like this is because of the bar body, because some cells, so this is the mother cell, and this mother cell can produce two daughter cells through the mitosis when the cat grows, right? And okay, so now two daughter cells are identical in terms of the genotypes. However, in these two daughter cells, one X chromosome will be randomly selected to be suppressed, to be repressed. So what's the consequence? So for example, in this daughter cell, if this X chromosome, the X chromosome with the lowercase b has been selected for repression, then only this allele will be expressed. That will gives you the black hair, right? However, if you look at another daughter cell, so if in this cell, the X chromosome carrying the capital B has been selected for suppression, and only this lowercase b allele has been expressed. So this will be the phenotype showing the orange hair. So that's why you're going to see, so this area, you're going to see one type of hair color, and this area, another type of hair color. So this is because of the bar body. It is a very special mechanism for the dosage compensation in mammals. Okay, so this is the standard situation, but can anyone tell me, so in a very, very rare situation, a calico cat could be male. So why is that?
It would be because the female has the non-disjointed XX chromosome, and the male would donate a Y chromosome, since the presence of the Y chromosome dictates maleness. Very good. Yeah, very good. So this is one possibility. So the female parents, so this is one possibility. So the non-disjunction occurs in female, right? The female will produce these gametes. And now the male will produce a gamete with Y chromosome. This come together to form this cat. And since this cat has two X chromosomes, all right, the bar body will produce this kind of color, right? And there's another possibility, like, so the non-disjunction can also occur in male, which means like the male cat can produce, for example, this gametes, and the female can produce this gametes, and this come together to form a zygote of this, right? but this situation is very rare. All right, very good. All right. Okay, any questions here? Okay, very good.
I guess the main thing to keep in mind is that in mammals, the genes aren't hyperactivated per se to compensate, correct?
Yes, so those genes must be on the X chromosomes, right? So right now we are talking about the genes are on X chromosomes. So if the genes are on autosomes, there's nothing we need to worry about the dosage compensation in male and female, right?
Okay, yeah, I understand. Yeah,
Yeah,
I was talking
okay.
about the previous slide.
Right.
Thank you.
Sure. Very good. All right. So this is a summary slide. So I'm gonna, move on this. All right. So now we're gonna talk about some other sex-related inheritance pattern. The first one is X-linked recessive phenotypes. So in this case, okay, so if there is a affected male, because this is X-linked, okay, there's only one copy here, right? The affected male individual will pass this allele to his daughter, but his daughter may be only a carrier. So this phenotype will not be affecting these two daughters, but these two daughters carries one allele, this diseased allele from their father, and this allele can be further passed to the grandson. So this is the feature that you can identify this disorder from the pedigree trees. All right, those are the features for the X-linked recessive phenotypes. All right, and X-linked dominant phenotype. So in this case, oops, the affected male will pass the diseased allele to his daughter, and because this is a dominant X-linked dominant phenotype, so these two daughters will be affected by this disease as well. And so when this one daughter has kids, so each kids, no matter what type of gender, so they all have equal chance, 1/2, to be affected by this disease. So this is the feature that we can identify from the pedigree tree. So this is for the X-linked dominant phenotype. All right. So for Y-linked inheritance pattern, it's easy because this disease or these phenotypes is only passed through males in the pedigree. So only males will be affected by this phenotype.
What are some examples of Y-linked inheritance, like diseases?
That's a good question. So I cannot give you an example right now, but yeah, I will look for example.
Okay, thank you.
I'm going to show you.
Okay. So
But
I'm assuming in the same vein, we won't need to know the specific diseases and their specific inheritance traits or patterns?
No, so I will not test you on that. So, but sometimes I will give you a pedigree in the test. So you need to figure out. So based on this pedigree, what is so the possible disease type? So you need to figure out. So for example, if I give you a pedigree tree like this, so immediately you can say, okay, because this phenotype or this disease only affected the males in this pedigree, so this must be a Y-linked inheritance disease, right?
I understand. Thank you.
Sure. All right. So yeah, okay. So I will look for some examples for the Y-linked inheritance disease. Okay. Next time. Let me ready to. Okay. So this is another phenotypes. So the genes controlling this phenotype is not on X chromosomes or Y chromosomes. Those genes are located on autosomes. However, the expression of those genes are limited by different sexes. So, for example, let's look at this example: the milk production in cows. All right, there are one gene, two alleles. This gene is located on autosome, not X chromosome or Y chromosome, and the capital L is dominant to the little l. So the capital L will give the lots of milk phenotype, but the little l will gives a cow a non-milk phenotype. However, this phenotypes only shows the variation of the phenotype only show in female, not in male. Although in male cows, they also have this autosomal genes, right? But so the genotypes will not produce any difference in the phenotypes. These genes, different alleles, only produce variability in the phenotypes in female. Okay. So in this case, we call this is the autosomal sex-limited phenotypes. Right. So another type is called the autosomal sex-influenced traits. All right. The same, these genes are located on the autosomes, not on the sex chromosomes. All right. For example, in human, there's one gene in autosome. This gene controls the bald or non-bald phenotypes in both male and female. All right. So if the genotype is B prime and B prime, this gives bald phenotypes in both male and female. So if the genotype is B and B, this will give both non-bald in male and female. However, when we look at the heterozygous genotype, so the B prime and the B, so the dominance and the recessive relationship is totally different in male and female. So that's why we call this autosomal sex-influenced genes. So for example, if for a heterozygous genotype, in male, this genotype will give you the bald in male, but non-bald in female. All right. So this is another mechanism. Okay. Another type is the maternal effect inheritance. So in human, as we know, the nuclear DNA, half of the nuclear DNA came from your father, and the other half came from your mother, right? So which means like a nuclear DNA will be influenced by both parents. However, there's some other DNA materials in your cell, so mitochondrial DNA. So mitochondria also has the DNA, and those DNA are only inherited from your mom. So that's why some phenotypes that are associated with the mitochondrial DNA, that's totally maternal effect inheritance pattern from your mom. Okay. All right. So so let's look at the examples showing the maternal effects. So first, why maternal effect will take effect? Because so when the maternal effects is taking effect, which means like the genotype of the individual, of the kids, is not important. So no matter what kind of genotype the kid has, it doesn't matter. It only this phenotype is only determined by mom's genotype. Why is that? Because mom provides something in the cytoplasm, such as protein, that can determine the phenotype of her offspring. So let's look at this example here. All right. So there's one gene, D, the capital D will show the phenotypes of the right coin, so right coin. And the little d will gives you a different, the recessive phenotype, left coin. All right. So now if we cross this female, which is the big D, with this male having the little d, so this gives you all right coin. Why? Because the phenotype of the F1 progeny is not depending on their own genotypes. So their phenotype, the phenotype of the F1 progeny only depend on the genotype of their mom. All right, that's why everyone here shows the right coin, right? Right coin. All right, let's look at this cross. So we just exchange the role of the two parents. Now we cross the female with little d with the male with capital D and capital D. Then the genotype of the F1 progeny in this cross is the same, heterozygous. However, as I mentioned, this is a classical maternal influence inheritance pattern. So the phenotype of this progeny does not depend on their own genotype. Those progeny, the phenotype of those F1 progeny will depend on their mom's genotype. So their mom's genotype is this, because the mom's genotype determines the protein in their cell, in their cytoplasma, right? Those protein will determine the direction of the coin. So that's why in this cross, all the F1 progeny shows the left coin. Is that interesting? All right. Now, when we do the intercross between this F1, the entire F1 progeny, so what happens here? So everyone of the F2 progeny will show the right coin. So why is that? Can anyone tell me?
Is it because the female passed on her little d?
You mean the female in F1 passed down the little d?
Yes.
Actually, so to answer this question, we also think about the maternal effect of the inheritance pattern, because so the phenotypes of this progeny, right? The phenotypes of all this F2 progeny will depend on their mom's genotype. So their mom will come from this progeny, this generation, right? Look at the genotypes of this generation. They are all big D and small d. Do you agree?
Yes.
So what's the phenotype from this genotype? The right coin, right? Is that right? So since,
Yes.
yeah, so their mom, so okay, so those F2 progeny, their mom, so their mom's genotype is big D and small d. So this will determine their phenotype. So they are all right coin. So that's the answer for this question. Does that make sense?
Yes, that makes sense.
Okay. So now, so let's look at the third, the F3 population progeny. So when we did the intercross among this F2 progeny, so why some left coin phenotype reappeared here? So why is that?
Is it because we have the homozygous recessive sample, and so since one of the mothers has the homozygous recessive genotype, it would pass on that phenotype to its offspring?
That is right. Yes, that is right. So there's the same thing, because so when you look at the phenotype of the F3 progeny, right, we need to look back to the F2 generation to see what their mother's genotype could be, right? Because their mother's genotype, some of them could be this, and some of their mothers could be the little d homozygous, right? if there are some of the progeny's mother carrying the genotype like this, the little d, this will determine their phenotypes in the F3 generation. Is that right? Yeah. Okay, very good. Any questions here? Okay, so I think this this also shows you the maternal inheritance pattern. So in human and in mammal, in animals, we inherited some genetic features from mother through the mitochondrial genes. In plants, this is the cell organelle that can carries the genetic information from mom to the next generation, the chloroplast. And so this just shows you an example that many human conditions that are associated with the mitochondrial mutations are passed through our moms. Oops. Okay. Okay. So this is an example showing that the maternal effect inheritance pattern in the plants. So this is a very interesting example. So let's look at this this plant. So if those plant the color of the leaves are totally determined by the mitochondrial color, right? So those are little mitochondria here. If the mom's egg only has the white mitochondria with not mitochondria, sorry, chloroplast. The chloroplast without the color, so this will gives you the white color leaves, right? So if the mom's egg only has the chloroplast with the color, the green color, this will gives you only the green color leaves. However, if the mom's egg has a mixture of the chloroplast with and without color, so this will gives you the leaves like this, the variegated leaves. So it's an interesting genetic pattern here. All right, I think this is all for today. So we have like maybe 10 minutes for questions. So do you have any questions that I can help answer now? And next week, on Tuesday, we're going to have our first midterm. So today and tomorrow, I will work on the exams. Also, I'm going to put a practice test onto the iLearn. So if you have time, please do the practice test, because the practice test will give you an idea. So what kind of like problems will show up in the real midterm next week. So on Friday this week in the afternoon, I have an office hours. So I'm going to extend my office hours to 2 hours. And so in case you have questions in the practice test, so we can discuss those questions to help you prepare for the coming midterms.
Hi, Professor. Oh,
yeah.
Go for it. You can go.
No, I'm done. Thank you.
Oh, okay. Hi, Professor. So I just have a quick question.
Mm-hmm.
It's about the non-disjunction, the divisions for the second and the first. Yeah, that one slide. Can you please repeat one more time how exactly the chromosomes are like divided again?
Sure. Okay. All right. So let me show you the process, the standard process for meiosis. Okay. So this is a homologous chromosome pair during the meiosis, right? Okay. So in a standard situation, in the first division, so you're gonna see this two will separate, right? So the spindle thread will pull these two homologous chromosomes apart. So what's the product is here? So if everything is normal, you're gonna see one chromosome in one daughter cell, and the other homologous chromosomes in another daughter cell, right? So this is the first division. Now the second division, this one will produce two daughter chromosomes. And so there's one sister chromatase and the other sister chromatase. I label this one as 1 and 2. So in this cell, we have 3 sister chromatase and 4 sister chromatase, right? So if everything goes normal, so these 2 sister chromatase, 1 and 2, will end up in 2 different daughter chromosomes. So this gamete will have 1 here. This gamete will have 2 here. The same thing for this cell. This gamete will have 3. This gamete will have 4, right? So this is the standard. Okay, now let's see if non-disjunction occurs at the first division. So these two do not separate. They come together, see? They come together into this cell, then leaving nothing in this cell. Is that right?
Yes. Okay.
Okay. Now, so this is how the non-disjunction occurs in the first division, because these two homologous chromosomes did not separate correctly, right? But in the second division, so this one and this one will separate. So this one goes to here, and this one separates. So this one goes here, and this one goes here. This one goes here. But nothing happened here. There's no such kind of chromosome in this cell, right? So you're gonna see the n+1 gametes for this one, n+1 for this one, but this is n-1 because one chromosome is missing for these two gametes. All right, so let's look at the second situation. So for the first division, everything is okay. So these two homologous chromosomes separate and they end up in 2 different daughter chromosomes like this. Right? This stage is normal. So this stage is okay. However, at the second division stage, so this one has non-disjunction events here. So normally, these 2 sister chromatids should separate, end up into 2 different gametes, but for this one, it didn't. So they come together, these 2 sister chromatids come together into one gamete and leaving the other gametes empty. Nothing here, right? But this one is normal because this one, the 2 sister chromatids separate in a standard way. And you're gonna see 2 normal gametes here, but 2 abnormal gametes here.
Okay, perfect. Thank you so much. That clears up everything.
Sure. Any other questions?
Hello, yes, can you hear me?
Yeah, I can hear you.
Yes, so I know you mentioned that the non-disjunction is incredibly rare, but
Mhm.
I'm curious, why doesn't the cell recognize that something is wrong when it does happen?
Yeah, that's a very good question. I think the reasons could be many. Is because the cell is doing a lot of things following the routines. If something goes wrong, it could lead to the abnormal consequence, right? But cell also has some mechanism to checkpoint to make sure everything is going on according to the right direction. But why is this? I think this might be related to some genes, is because the meiosis process is also determined by a lot of genes, if which are involved in a biological pathways. If some genes goes wrong, probably this will happen. But which gene goes wrong, we don't know. Maybe different individual has different problem with those genes in the pathway. But this is a good question. So if you are interested in that, maybe you can look for some explanation evidence from Google. I hope you can find anything. If you cannot, let me know so we can work on this together.
All right, thank you for the information.
Sure. Any other questions here?
Professor, I have a question regards the exam.
Mhm.
So the best way, like, to be prepared, just the lectures and the homework. There's anything else we can do?
Yes. So definitely review the PowerPoint slides and the reading assignment and doing the practice, doing the assigned homework. Yeah, that assigned problems that help a lot for preparing for the midterm. And also, I'm gonna, as I mentioned today or tomorrow, I'm gonna upload a practice exam. So yeah, doing the practice exam will definitely help you. Yeah.
Great. Thank you so much. So it's going to be next Tuesday, right?
Next Tuesday, yes.
And Professor, if you could go into detail about the format of it, like
For what? I'm sorry?
for the midterm next
For
week.
the midterm? Yeah.
Like, what would be the format of it?
The format, I think for both midterms, I'm gonna use the same format like the quiz. I'm gonna give you like several questions. You're gonna give me short answers to each of them. Yeah, that's the format for 2 midterms. But for finals, I think, so I'm gonna follow the previous routine. For finals, I'm gonna only give you the multiple choice questions on the finals. Did I answer your question?
Yeah.
Okay. Any other questions? Okay. All right. If you do not have questions, so let's stop here today. Thank you.